So we started our we ended our last class wherein we discussed about all the steps involved in the maturation of uh, B cell. So we discussed that there is one antigen dependent phase, and then there is something called as the antigen independent phase. So we'll further our discussion from there. If there's any doubt that you all have encountered from the last session, we can also take up that first.
Recording in progress.
Okay. So for today's class, we'll try to first finish up our discussion about the entire process of B cell activation matching. So we ended our class with B cell proliferation. Just so sorry for the disturbance. Okay. So we stopped here where we were discussing about the process of maturation and activation of B and T cells. So we first begin with the discussion regarding the B cell. And we saw that there is a phase called antigen independent phase, which we saw that there's some sort of rearrangements that are happening. Sometimes it's a heavy chain rearrangement, then we have light chain rearrangement. Ultimately, we have some antigenic commitment, and the naive B cells can now get into the peripheral lymphoid organs, wherein they are supposed they are where they are now called to be mature B cells. These mature B cells have a particular immunocompetency, which means that they can recognize antigens of a particular make. And whenever such an antigen is encountered by them, in presence of certain desirable cytokines released by certain other helper cells, they are activated. Naval, can you please mute yourself? So that they are activated. And once they are activated, they will undergo clonal selection, clonal expansion, and something called as class switching. So we'll understand all of these things one by one. So as far as the previous discussion was concerned, we just have another representation from the book itself, the QB, where they very clearly show that there's a bone marrow, and they have also mentioned that there is a marker. So in immunology, if so far in all the discussions, by now you have realized that every particular cell has certain markers on its surface, and these markers are actually used for designating the cell to a particular cell type. We have the progenitor B cells, which express this very important CD45R, which is a surface marker, also called as B220. In essentially, it is nothing but a transmembrane tyrosine phosphatase. So, as the name suggests, it must be having certain tyrosine residues, wherein some phosphatases might bind, which means the residue essentially might be having a phosphorylation. Phosphatase, there must be certain enzyme acting on this phosphate residue, which is going to dephosphorylate it. And dephosphorylation and phosphorylation is a very important, we'll discuss in today's class also. It's a very important way of signal transduction, right? So this is essentially the same thing that was happening in the peripheral lymphoid organ. They have discussed it further. One thing that is important is we were talking initially in the previous lectures about selection. Selection for autoreactive antibodies, which means the B cells that can react to our own body's antigen and that can have detrimental consequences. So they need to be selected or filtered off. So one possible way for doing it is that if we have a pre-B cell, which is having a light. So all these rearrangements are happening. If after all these rearrangement, we have a pre-B cell, which is essentially having antibodies that can now react to the self antigen, they are selected. First thing is to pick them out. And the next decision is to whether uh, ask them to again undergo the process of rearrangement, say, so that the specificity of the antigen binding sites could be changed. So that is called editing. Or if that is not possible, if editing also again gives back an autoreactive B cell, they have to be triggered for death via cell apoptosis. So this selection and editing process is actually happening at the stage where a pre-B cell has to now become an immature B cell. I have also added certain markers for all the specific cell types that we have discussed in the entire process of maturation. These are certain things that you might want to remember for the purpose of certain competitive exams or something like that. You might also encounter some of these terminologies in the weekly assignments that you all have to appear for. Okay. And so one more thing, just an added, so we are just trying to enrich our discussion of that from the last class. So we have something called as the bone marrow stromal cells. So we have discussed stromal cells previously while we were discussing all the lymphoid organs, which is essentially the cells present in the matrix wherein the different cells are actually differentiating. So these stromal cells have quite a large surface area and they actually express certain cell adhesion molecules such as VCAM or something called as SCF over here. So all these, so the SCF is not essentially an adhesion molecule, but it is also a receptor which can binding of it to certain ligand which is present on your pro B cell can initiate certain signaling mechanism. So what is happening is that you have a pro B cell, which inside the bone marrow is actually getting to interact with these bone marrow stromal cells based on certain adhesion molecules. It binds to the cell surfaces. Based on next comes that once a cell adhesion molecule has bound, it allows for your receptor and ligand present on these two cells to come in near vicinity. Eventually, they bind and they trigger a signaling response. When this response is triggered, this B cell is now expressing a receptor for a particular cytokine called as the IL-7. So now this pro B cell, when it can respond to IL-7, because now because of this signaling, it is expressing the receptors, it is now responsive to IL-7 cytokine. Now, who is producing this IL-7 cytokine is again the stromal cell in itself. So these stromal cells are now going to produce IL-7, which binds to the pre-B cells, eventually leading to the transition of pro-B cells to the pre-B cells. And later, these pre-B cells can form immature B cells, which go to mature lineage. It then gains its antigen competency and everything that we have already discussed, right? Two other things that for now, just so that you know, because these words might come up in our discussion for today's class. Once we have a naive B cell in the peripheral circulation, so you might find these terminologies like the naive and mature variably. Some might say that naive B cell is what enters the peripheral lymphoid organs, and they might have some other text which is saying that the mature B cell comes in. So essential difference between naive and mature B cell is not much because all the other chains and everything is already in place. It's just the nomenclature that can vary from one author to the other, right? So once we have the naive B cell encountering the antigen in the peripheral lymphoid organ, it becomes an activated B cell, which means it will and it now has to undergo clonal proliferation. So in clonal proliferation, you have this one cell type producing exact copies. So it will now divide into exact exact copies of plasma cells. What is also possible is that while it is producing plasma cells which has the same specificity, the receptors on the plasma cells having same specificity for the particular type of antigen encountered, you can also have these cells demonstrating class switching. So class switching is a phenomena wherein the specificity of the antigen binding site remains the same. However, the constant region, which is actually determining that which type of antibody is produced, if you remember from previous discussion, the first antibody that is expressed on the surface of the B cells is the IgM. Along with IgM, we also discussed that IgD antibody is formed, but the exact reason for the production of these IgD antibodies bound to the cell surface while it is developing is not known. So people have tried to knock out IgD and they find that there is no effect on the overall that there is no effect on the overall development of the naive B cells into the mature B cells, right? So IgM is the primary antibody, which is very important and expressed on activated B cells. Now the plasma cells will also be having IgM antibody so that it can bind to the antigen. What happens to the plasma cells? That these IgM antibodies, which have a transmembrane domain, that transmembrane domain is lost, so that instead of being bound to the surface, they are now being produced in the secreted form. Apart from that, you can also have IgM antibodies changing into either of the other types of antibody, which is IgG, IgA, or IgE. Now, into which type this class switching happens, that is determined by the constitution of the cytokine that is present in the vicinity of this activated B cell. So we'll come to that part sometime later in the discussion, that to which type this is happening, which cytokine determines what. There's a very nice schematic and it's something that you need to just remember. But what you need right now need to remember is that this IgG, the antigen specificity of this IgG molecule is not different. So if I say that the antigen specificity, if I just give a shape to this antigen, the antigen specificity was somewhere triangular here. It's not that after class switching, it will now be able to bind to some other type of antigen. It will still be the specificity remains towards the same type of antigen. It is just the C-terminal domain that changes because of certain. So this is the structure of B cell receptor. We have the antigen binding sites on these two areas. So that is why it is antibody is called a bivalent molecule. All of them have the same specificity here. It's just something at the C terminal, the constant region, that is different. And when we read about antibody types further, we'll see if we can cover it in today's class. You would know that how exactly all these antibodies are different. And the reason, the requirement for class switching stems from the fact that all these isotype of antibodies, they are all having different affinity towards the same antigen because of certain interaction that the C-terminal domain of all these types of antibodies can mediate. So the effective, to increase the overall effectiveness of clearing out the antigen, for that purpose, the class switching is happening. That is what you need to remember for now. Another fact that happens is affinity maturation. So what is affinity maturation? So let's just say that once we have a triangular sort of antigen here, the antigen binding site of antibody might be something like this. So it is somewhat triangular, but it has something like a curved sort of end. Whereas this is a very pointed sort of antigen. This is just certain representative diagram that I'm drawing for you to understand that. In the process of affinity maturation, there will be very point mutations are going to happen. So how in the same cell, the next type of mutation is not possible. But while the cell is undergoing clonal proliferation, it is actually getting a chance to replicate again and again, and also to accumulate some mutations. So, point mutations of this antigen binding site is going to refine the specificity of this binding site to the exact similar nature of the antigen that it has encountered. So if you would notice that these two are not very different, right? Similarly, it might be that just one amino acid has to be changed for the affinity of this binding of antigen antibody interaction to be very, very high. So that sort of thing when it is happening, this is known, thank you. This is known as the process of affinity maturation. So affinity maturation and class switching are two phenomena that is happening while the antibody is undergoing, while the B cell is undergoing encountering the antigen. Once the initial encounter with the antigen is cleared off, we do have the memory B cells, which is going to be maintained throughout your life. And we have discussed this memory component already. Now, so far we were visualizing a single B cell having some receptor called which is the antibody molecule in itself. And along with that, we were discussing that we just made these two lines here to denote that there is something as Ig alpha and Ig beta molecule present associated with the B cell receptor, which in itself is an antibody molecule. Now, what exactly is the function of this B cell receptor? We'll look into that part, right? So if you look here, these Ig beta and alpha molecule are present in very close association with this antibody molecule. So what you also see here is the structure of an antibody. So I am sure that you all have already gone through the recorded lectures and you are familiar with this structure. If any one of you seem to miss out on some information, because those are very straightforward, factual, detail based topics. So we'll try to discuss the concepts here and will not get into the lot of facts that can be simply read about and memorized. We'll focus on the understanding part here so that your doubts are clear about the subject. So if you recall from the recorded lectures of some other weeks, maybe, we have read that antibody molecule, it's a Y-shaped structure. But what we were so far drawing like this Y molecule, there essentially is so we have one chain here. This is one part, which is known as the heavy chain, and this is the other heavy chain. With this heavy chain, it's not really a very straight line structure that forms when the protein folds in 3D. You will see that there are some loops that are actually forming while the protein folding is happening. Of all, so these are known as, I think we call it as CD1, CD2, CD3, CDRs. So these are CDRs, right? And we just name it from as CD1, CDR1, 2, and 3. Most important is this N-terminal region of the antibody molecule, because it has a lot of variance in the sequence. And if you would again, if you further remember, almost the majority of the variance is actually restricted to three regions on the antibody molecule known as the hypervariable region. What does this mean? This means that so if I just grade in terms of, so if we look from this terminal of antibody, so the C terminal, which is actually also embedded inside the plasma membrane. So this from the C terminal towards the N terminal, in some way, the variability increases, right? So we have this CDR, which is the most conserved compared to the variable region here. So this region is the most variable part, and inside of this region also we have three such region which seems to have the maximum variability. So these regions in together are called as the hyper variable region. Similarly, on the light chain also, we have three hypervariable region. I'll just write it here. And together, when these two hypervariable region of the C terminal of the light chain and the heavy chain, they come together to indeed form what is called as the antibody antigen binding cleft. So it is a cleft because it essentially is in the shape of a furrow where an antigen can come, and based on its complementarity to the sequence of these hypervariable regions, the antigen can either bind to it or it might not bind to it. Now, if you see, once the antigen binding has happened, so that is just one event in the overall process of activation. Again, look back, we have a mature B cell which can bind antigen, but it has to get activated. So what means upon activation is clonal proliferation has to occur. So so far what was maintained as an arresting G naught phase, or maybe when it has come into the peripheral organ until the time it encounters its respective antigen, it is in a dormant G naught. It's in a resting phase. Once it has to so activation essentially overlaps with clonal proliferation, and for the cell to undergo proliferation, it needs to divide. Now, what the binding that has actually occurred on the antibody molecule bound to the B cell receptor, who will transfer this signal to the inside of the cell? Because the C-terminal domain of antibody here is very, very short. It's just 3 to 4 amino acids. That job is essentially performed by the Ig beta and Ig alpha molecule, which is present in very close vicinity of your membrane bound antibody. These two complexes together constitute the B cell receptor. So if you see here, we have the Ig beta molecule here has a 48 amino acid long tail, and the tail for Ig alpha is even longer. It's somewhere around 61 amino acid. So that much we'll further see that these tails also have certain conserved region where some tyrosine molecules are present. Tyrosine again is going to either get phosphorylated or dephosphorylated. So essentially the tyrosine attached here is in a non-phosphorylated state, and binding of by the action of certain kinases, a phosphate addition is going to happen that is going to mediate the entire intracellular signaling event. So when the signal is triggered, you will have the cell would now know that it has to say undergo proliferation, or maybe it has to produce secretory antibodies and all those things, right? So just allow me 2 minutes. I'll be back. You can have a look and read the slide in the meantime. Okay. So if you could just go through it. So essentially your B cell receptors are transmembrane protein complexes which firstly will be of the IgM make right here. And we will have certain disulfide molecules linking all these different polypeptides together, right? There are also some additional molecules which are present on the surface of the B cell membrane, and they are equally important. So a B cell receptor is a complex of your immunoglobulin, the membrane bound immunoglobulin, and Ig alpha and Ig beta. Along with that, we have something called as the CD19. So the role of CD19 is in the process of uh, it facilitates the it facilitates the binding of antigen to this immunoglobulin molecule. I might show you a diagram for you to visualize how that is happening. So it basically kind of gets to hover over on this antigen molecule, and we have this antigen here. This facilitates this antigen's very close association to the antigen binding cleft. So this, along with CD81, and which is also called as TAPA1, and CD21, also called as CR2, these molecules together constitute the B cell co-receptor. Along with that, we have the class 2 MHC molecule present on the surface. Apart from that, we also have a co-stimulatory molecule known as the B7 molecule, which we will see is a very transfers a very important co-stimulation to the T helper cells, which will eventually come and communicate with the B lymphocytes, right? So, when this B cell is activated, the number of this B7 and the class 2 MHC molecule is upregulated multiple folds. And this in turn, the interaction of this B7 to the CD28 molecule of the T helper cells is going to further activate the T cells, which is now going to release the cytokines that can determine the fate of the B cells. Apart from that, we also have some adhesion molecules, because adhesion molecules will be required because the cell might have to get marginalized when the interaction of the B and T cell is happening. So the cells need to be first need to come together, stay in the near vicinity, which is a faster process, and that holding of the cells together allows for all these other receptors to bind to. If you look here, we have the much discussed CD40, which is bound to the surface of the B cells, and we will look into that how it is helping. So while I was reading about the interaction of B and T cells for this particular class, the discussion lands very clear. We do have the CD40L molecules on the T helper cells, and we will just come to that in a couple of minutes, right? Okay. So apart from that, what I also wanted to bring up that in the all the cells of involved in the immune system, they are very nicely decorated with different super families of immunoglobulins, or maybe the globular protein structures, right? So if you look here, your Ig alpha beta, they also have this. So if you just look at the entire picture over here, you will see that almost all the different molecules, the different receptors or accessory proteins, some adhesion molecules that are present on these cells, they are all almost of the similar make, and they are all the members of the immunoglobulin superfamily. So they are very structurally related groups, and all these, so we have CD3, which we will very nicely discuss when we are talking about the T cells. So this is the T cell receptor that we have briefly talked about. We have also talked about MHC molecules, and there are two types of it, the class 1 and class 2 molecule. If you look here, even they have the similar structural features, and similarly the adhesion molecules that part of the overall in that facilitate this overall interaction, they are all belonging to the immunoglobulin superfamily. Okay, so now we'll come to the process of B cell activation. Just a minute. Okay. So the process of B cell activation. So far we were discussing in detail the process of B cell maturation. Now we'll move to our discussion of activation. So activation wherein a mature B cell has already encountered an antigen. So the first thing first, it needs an antigen, and this antigen can be of two types. So essentially your antigen is can be of multiple types, but based on what is the reaction that follows, different types of antigens have been classified into two broad categories. One is your thymus dependent antigen, wherein a T helper cell is directly involved, which means that there is going to be a direct contact of the T helper cells and the B cells. So, and if I talk about thymus independent, so on that's exactly the opposite. So this is T helper cell independent, and why we are calling it thymus? Because essentially the T cells are coming from thymus. So people have actually performed thymectomy, and they have seen that which kind of antigen is uh, responded to or the body fails to respond to, and based on that they have given this sort of name. But in thymus also, if somehow the TC cells and T helper cells could be specifically depleted, we would see that the response is actually T helper cell dependent, right? Or T helper cell independent. So T helper cell independent response means that the direct participation of the T helper cells is not required. However, the cytokines that are produced from the T helper cell might be required for one of the two further classification that is performed for TI. So TI and TD antigens. So TI antigens are further typed into type 1 and type 2. Type 1 is essentially for the lipopolysaccharides, which is present in the bacterial cell walls. And what happens that if a LPS stimulus is present, if LPS antigens, maybe the bacteria has been lysed or we have a bacteria and the uh, B cell recognizes it based on the lipopolysaccharide component of the cell wall, there is a polyclonal activation of B cell. What does this mean? So far we were learning about clonal selection, where the B cell of a particular type is activated, it's getting selected, it gets to proliferate, whatever. But here, if one B cell encounters a lipopolysaccharide, so what happens is your entire bacteria, it's a huge structure, and different regions of the LPS, which is the cell wall of the bacteria, can actually be recognized by multiple B cells, each with its different specificity. And that is how polyclonal. So different types of B cells are produced, and people have quantified that almost 1/3 population of the entire B cell repertoire that is present in the body can get activated in the presence of a type 1 TI antigen, particularly the LPS. Type 2 TI antigen. So it is being constituted by polymeric protein or cell wall polysaccharides. So essentially, whenever we have similar uh, similar sequence of amino acid is repeating, or we have two proteins which is multiple time linked together into a polymeric association, or we have multiple saccharides of the same kind which is repeated. So any sort of repetitive structure of this biomolecule is classified under the TI type 2 antigen. Over here, the type 1 antigen, type 1 TI antigen, this response is completely independent of T helper cell. So we only have the interaction of type 1 TI antigen to the antigen binding site of the B cell. We have polyclonal activation, and the antibodies that are produced are going to take care of the antigen. The type 2 TI antigen requires, so how it happens? So these structures are actually binding to this Y-shaped molecule, and it's a huge structure. So maybe two closely associated molecules can come together and get cross-linked. So I'll just show you a diagram. So this is what cross-linking looks like. Because so we were thinking that we have one B cell with one antibody molecule. It's not like that. We have multiple antibody molecules, all of them of the IgM type, present in the near vicinity together. When an antigen is binding to two antigen binding sites of two different antibody, what happens is known as cross-linking of the B cell. Whenever there is cross-linking, so when this type 2 antigen binds to B cell, there is cross-linking of the IgM receptors bound on the B cell. And further, there is the requirement of T helper cell derived cytokines. So here cytokines were completely not required, but after cross-linking, what is the fate that follows to take care of the antigen? That is determined by the cytokine that has been produced by the T helper cells. It is also important to know that in thymus independent antigen, no memory component happens, and that there is no class switching of antibodies. So this is essentially just a single one-time response that the body mounts. There is no memory that the body recalls whenever the same sort of antigen is again encountered, and there is only IgM in secreted form maybe, which is taking care of the entire situation. There is no class switching that happens. And so that's something that there are two types of antigen that you must know. And here we have a table that summarizes the property of these two types of antigen. The chemical make of thymus-dependent antigen is actually proteinaceous in nature, whereas the TI antigens, they can be cell wall components or polymeric proteinaceous antigens present on the capsule, or say the flagella of a bacteria or a protozoa, those kind of structures. Humoral response, we do have a humoral in terms of humoral response, we have isotype switching in TD antigen, but there is no isotype switching in type 1, and there is very limited isotype switching in type 2 TI antigen. There is affinity maturation for TD antigen, none for the TI antigen. We have immunological memory forming in TD antigen, but no memory for the TI antigens. And in polyclonal activations over here, there is clonal selection, that is only one type of antibody is produced, only one type of antibody with a specific specificity is only produced. We have a lot of polyclonal activation here, only in type 1 of TI antigen. So polyclonal activation is a particular, is a feature for your type 1 TI antigen, because even type 2 TI antigen do not show polyclonal activation. Now, once you understand that the process of B cell activation requires antigen, we have two types of antigen based on whether it requires your T helper cells or it do not require T helper cells. So two things are for sure. Antigen is required and there might be a role of T helper cell. Secondly, what we talked about is that the cell is right now in a resting state. So it has to get activated. Activated. So for the purpose of proliferation, clonal selection, it needs to undergo division. So for this trigger of the G0 cell to transform into G1 cell, we require a particular signal named as the competence signal. And for transition of a cell from G1 to S phase requires another signal called as the progression signal. So there are say two checkpoints that is actually allowing the cell division to occur. So because you must understand that cell division is a very tightly regulated process, because unwanted division can cause more harm than good inside the body. So there has to be certain checks, or there has to be certain chemical cues or certain signaling mechanisms in place that ensures that your cell division is required right now, and only then the cell undergoes division. So over here, this G0, this G0 to G1 transition is under the control of what we call as the competence signal, and we'll see that what exactly is the competence signal forming here. And if we see that, then again, competence signal also is a two-step process. So it is called as signal 1 and signal 2. Again, what is the signal 1, what is signal 2 can differ for your TI and TD antigen. So what you need to understand is that different antigens can actually elicit different cellular responses, different cellular signaling mechanism, based on which your downstream events will be determined. For progression signal, the G1 to S phase transition, this is similar for your TI and TD antigens. Both of it based on whatever is the cytokine that is present on the near vicinity, and or the ligands that are involved, the ligands that is going to, that gets to bind to the B cell membrane receptor. So when I'm saying B cell membrane receptors, we're essentially talking about all these cell receptors. So all based on whatever is the this interaction, we have the progression signal happening. Once the cell reaches S phase, it is now going to definitely transition to G2 phase. It is going to complete the M phase, and eventually we have two daughter cells forming out of one progenitor cell, or maybe the parent cell. So now if you look here, this is just a schematic taken. So for T1, TI1 antigen, which is they're referring to LPS, the binding of type 1 thymus independent antigen provides this signal for provides both of these signal, the signal 1 as well as signal 2. So signal 1 and signal 2 are essentially two chemical events happening inside the cell that is instructing the cell to get into the next phase. But for TD antigen, we have to have an antigen that gets to cross-link. This cross-linking provides the first signal, followed by the processing of the processing of the antigen antibody complex that happens further. We have the CD40 and CD40L molecule present on T helper cells. This antigen has to be presented to the TH cell by the MHC complex receptor, and the CD40 and CD40L binding. This provides the second signal that we have discussed. So both this signal as well as this signal, the cross-linking and the CD40-40L interaction, both of them together allows the transition from G1 to G0 to G1 phase. Okay. So once we understand that, but primarily most of the antigen that the body responds to are proteinaceous in nature. So most of our discussion, the generalization of the discussion is is like that for your TD antigens, right? Now, what is this signal that we are talking about? So signaling in the immune system is predominantly by the mechanism of phosphate. So either something is going to add a phosphate or something is going to remove a phosphate. Over here, we were talking about the Ig alpha and Ig beta molecules. We have protein tyrosine kinases, which are associated to the Ig alpha beta molecules in the PCR complex, and they mediate these signals. We'll just look into it, right? Okay. The first thing that happens is we have an antigen. This antigen comes and binds to of the antibody molecule present on the surface. Once this cross-linking, so this cross-linking, it generates signal 1, and in signal, because of this signal 1, what happens is we have increased expression of MHC class 2 molecules, and here we also have increased expression of B7 molecules. If you remember from here, we have the B7 molecule, which is required for providing a co-stimulus to T cells, right? So this cross-linking is generating signal 1, which is doing two things. Once this cross-linking has occurred, here we also have these Ig alpha and Ig beta molecule, which initially was non-phosphorylated. But when this cross-linking occurs, these ITAM regions, so ITAM sequence is where a lot of tyrosine residues are present, which can be phosphorylated. The tyrosine residues of the ITAM region, they can now they get to interact with the members of SRC family of tyrosine kinases. So just some names. So SRC is a family of tyrosine kinase. Essentially, kinase is going to add a phosphate. Because of this cross-linking, they are getting to interact with SRC family of tyrosine kinases, and two phosphate. So phosphate molecules are added to the tyrosine residues in the ITAM sequence. So this is the SRC kinase that gets stationed when this cross-linking happens. Once this phosphate has bind the ITAM molecules, it creates, so any sort of phosphorylation is going to bring some sort of change in the overall protein structure, and because of that, we have another kinase that gets to bind here, right? So this is called as the SYK kinase. So another kinase binds here, it gets to dock here, and based on that, we have multiple secondary messengers that can be produced in response to this binding. Which messengers? It can be the IP3 messenger. So there are essentially two pathways for signal transduction, which can be mediated by IP3 and DAP. In this pathway, there is a release of calcium intracellularly, or there can be an activation of PKCs. The other possible way is that there are G proteins that are getting activated, and we have RAS and RAS. So these are something which is part of signaling mechanisms, and we'll not get into the details of this. But it produces certain secondary messengers. So now this extracellular signal is transferred inside the cell, and within the cell we have secondary messengers that have been produced that can cause some change inside of the cell, and your signal from extracellular surface is communicated intracellularly. This can ask the cell to actually say, now we have to say divide, and in this process of division there can also happen some change in the gene expression pattern. So signal translocation occurs to nucleus. This activates certain transcription factors. Based on which transcription factor is produced, you can have transcription of some specific genes that come into play. Okay, so we have understood that how With this, we understand that how the extracellular signal is coming into the inside, is communicated to the inside of the cell. But what are the exact steps that follow? So we say that signal 1 was transferred here. Draw the diagram separately. So we have the B cell here, the antibody molecules present here, and it has now been cross-linked in presence of an antigen. This gives signal 1. In presence of signal 1, we have class 2 MHC molecule now expressed in higher numbers. So your cell is activated. We have class 2 MHC molecule. So MHC2 is constitutively expressed on the surface of B cell, right? So it is not like it was not there and only when the signal comes, but the expression of MHC class 2 increases. What also happens is the expression of another molecule called the B7 molecule increases. Eventually, eventually this antigen antibody complex formed here is going to get endocytosed. We have discussed it in the process of antigen presentation and processing that extracellular antigen can get endocytosed, and in the endocytic compartment it gets a chance to bind the MHC class 2 molecule, and the peptide, the processed peptide is presented in complex with the class 2 MHC molecule. Now this class 2 MHC molecule has to present it to a T helper cell. Again, apart from that, we just saw that there is something called as CD40. So another molecule present here called the CD40 molecule stationed here. And we have, when all of these receptors are expressed on the surface, whichever T helper cell is present in the near vicinity. So we have a T cell. Just drawn in too far. So we have a T cell coming in here. The T cell has TCRs. So this TCR is going to recognize this antigen present in complex with class 2 MHC molecule. Apart from that, this B cell has CD28 molecule, and it also has CD40L. So when all these three interactions are in place, this is so this is going to also send back, this binding is going to send back signal 2. So MHC comes first. We have the MHC coming first. We also have the B7 coming at the same time. When these interactions happen, this is followed by the interaction of CD40 and CD40L. CD40-CD40L interaction sends back signal 2. In presence of signal 2, we now have the B cell. So this interaction, let's just draw it like this. This interaction activates T helper cell, which produces cytokines. This also signals the B cell to produce cytokine receptors. Right? So this sends back cytokines when this interaction comes in, and this expresses the cytokine receptors now, which can be bound by the cytokine producing from T helper cell. So now if you see this, it is essentially one cell helping the other to help its own self in return, right? So it's a lot of crosstalk between the cells that are happening in the entire immune system, and we have certain chemical messenger that is facilitating the entire process. Here we are calling it as the cytokines, right? So this is, in summary, this is what is happening for the process of activation of B cells. If any one of you have any doubt for whatever has been discussed so far, you can please unmute and I'll just check if I am missing out on anything from the textbook. Okay. So here, so just to add on, once this is happening, so we now have the B cell. This is known as activated. Where is this activated B cell? So if you so you have to always link the concepts together in immunology. These activated B cells are present inside the germinal center that we have discussed while we are talking about the lymphoid organ. So we have this entire process of proliferation, activation right now is happening in the peripheral lymphoid organ, and where in there? It's in the germinal center. So it's within this germinal center that the immunoglobulin genes is. So maybe this cell now becomes a plasma cell, or it undergoes class switching, or anything that happens. It is all happening inside the germinal center. So receptors. Okay. So that is about the process of B cell activation. We can now get into our discussion with the different type, because now we have talked a lot about antibodies, this different structures of the antibody. So we'll just also try to quickly discuss about what is exactly the different types that we were just naming by so far. We'll move very, very quickly here so that we can stop by some time and then we can take our doubts if you all have any. So we have briefly discussed our structure of antibody. We'll just open this only as a reference diagram here. If you see here, among all the domains, so this is these. No doubts from any one of you so far?
No, ma'am, it's clear.
Okay. Okay, just ignore this bit for a while. We'll just, I could not put another diagram over here. I forgot to put one and will not waste a lot of time in drawing it. Okay. So here, if you see, so this we said is the antigen binding site. This part is known as the fragment AB, the antigen binding fragment. And this part is known as the constant region or FC. If you see here, so these regions, this is CH2, CH1, H2, CH3, CH4, and Sorry, this is take it. This is CH1, CH2, CH3, and CH4, and this is the variable region, right? This is a variable region of heavy chain and the other constant regions of heavy chain. Now, since we're talking about IgM, you would see that it has four such complementary complementary binding regions. Similarly, for the light chain, so you must know that this is the light chain, this is the variable part of it, and this is the constant part of it. Okay, so in here we have three such regions, which is known as the CDRs, right? CDR 1, 2, and 3. And if we talk about the different types of antibody now, am I not able to find a diagram for it? Okay, maybe we can just. So same as so IgM and give me a second, I'll find out the diagram, will be easier for us. Yeah. So if you look here, this is the most elaborate diagram that you can find for the structure of antibody. Essentially, if you look at it in 3D, no. So essentially, if you look at it in 3D, so what we're understanding as a two-dimensional structure here, this is how it looks in its 3D orientation, right? So just different sort of folding that comes in. But if you see that it's the variable regions that are always at the very tips of your 3D fold to allow easy binding and easy access for the antigen here. So this is the antigen binding site, and all the different chains and everything that we'll just discuss is folded like so. Here, if you look, so with this, we'll just quickly discuss that how the entire structural deduction for the antibody happened. If you see here, it's your IgM, which we're having four such domains. Apart from that, IgE is also having four such domains. If you look for IgG, so there are five kinds of antibodies to start with. IgM.
And the screen is not shared.
Okay. Can you see the screen now? Oh, I'm so sorry. This is what I'm saying, that when it folds in 3D, this is how the structure looks. If you just see here, this 3D, despite of the 3D fold, this is the Y-shaped structure that we were talking about. And your antigen binding sites, sites remain at the absolute tip of your Y molecule, despite its 3D folding in that fashion, right? So if we just quickly go through over it, it's IgM, IgD that appears first. Then when the class switching has to occur, we encounter IgG followed by IgA, and IgE is what is produced in very minimal quantities by the body under very particular reactions. But specifically when there is certain allergic reaction, that is when IgE has to come in. So if you see here, we have for IgM, for IgM and IgE, there's an additional domain which is not present for the gamma, which is essentially G, del, and alpha. So these are the Greek words from which we have these alphabets come in. Why have they been named so? So while the antibody production has to happen initially, so we can have, so there are two possible types of light chain that comes in. One is the kappa chain and the lambda chain. When finally, so this choice is there before the rearrangement process has to happen. So in the next class, we'll be discussing about the rearrangement process that happens. And so before this antibody molecule is assembled, there's two choice that the body has to decide from, that whether a kappa chain can be picked or a lambda chain can be picked. What is important that it is never such that we have one kappa chain and one lambda chain. Both the light chains are of the same make. So if it's kappa for both the chain. Similarly, so for light chain, we have two types, but for your heavy chain, based on which the isotypes have been named, we possibly have five such choices. One is the gamma chain, del chain, alpha chain, mu, or E, or epsilon chain, right? But again, both the heavy chains can only be of the same type. It is never that you will have one mu chain and one epsilon chains or all those random combinations, right? So apart from that, so the variable region we understood very well that this is your antigen binding region, but it's not like that this constant region has no role to play. These are the domains for biological activity. So if you remember from the last to last class, we discussed that how there are certain effector mechanisms that the antibodies can bring about. So we have something called as opsonization and neutralization, complement activation. All those things are actually being mediated by the FC, or the constant region of your antibody molecule. Okay, so apart from that, if you look here, we have a lot of multiple disulfide linkages that keeps all these different domains together, and also keeps these two chains together. So how people actually deciphered the overall structure of an antibody molecule is by actually mediating the cleavage, random cleavages. So different scientists actually used different enzymes, and then they see that whatever are the fractions that are formed, and based on which fraction can bind an antibody molecule, which fraction could not bind it, they have reduced the overall structure. And from there only, so FAB is actually fragment antigen binding. So when a cleavage was performed, so this was, so I'll just quickly mention, I'm sure you have encountered these. When papain digestion happens, the cleavage happens in this fashion. So this is, so red is your papain. Papain digestion gives two fragments. So you have one fraction that could bind the antibodies called the FAB fraction, and you could get a crystallizable fraction which is called as the FC, which the C that we usually say, we call that it as the constant region. But initially this name C comes from, because when the cleavage was performed, this fraction could be crystallized but cannot bind antigen. So the FC name was given to it. Similarly, there's another scientist who performed the digestion using pepsin. The pepsin digestion cuts over here. So pepsin, it produces a heavy molecule where two antigen molecules could be bound by the same fraction, and no FC. So this region could not be obtained. So FC fragments were obtained. They could not find a complete thing that can be crystallized, but some small peptides could be obtained. So they just named it FC fragments. So essentially, then they saw that which particular enzyme can actually cleave which segment of it based on these experiments. If the disulfide bond, so pepsin cleaves the disulfide bond below the disulfide bonds. We have a disulfide bond here. So this is called as the hinge region. Pepsin cleavage occurs below the disulfide bonds, and that's how you have two FAB fragments and no FC fragment was produced. At the same time, papain was cleaving at this point. So it was cleaving beneath a single disulfide bond, and that's why you have two FAB fragments and no FC fragment could and the FC fragment, because we still have these disulfide bonds which is keeping these two things together, and no degradation can happen. So that is how the structure of antibody was deduced. If you also look here, roughly the certain amino acid, the overall length of amino acids, the heavy chain is somewhere around 440 amino acid long, with this on almost all the different parts of it being of roughly of the same amino acid length. So the variable region, if you just do 440 by 4, it comes around somewhere 110 amino acid that constitutes the variable region for your heavy chain. Similarly, since your light chain is somewhere around 220 amino acid long, again half of this, so the variable region constituted by the light chain is also around 110 amino acid long. We have the disulfide bond that is definitely keeping these the entire protein together. But apart from that, there are also certain covalent bonds that is facilitating the interaction. And if you look here, so we have already discussed that we have these complementary determining regions that is also responsible for the biological activity. We have discussed about what are the different classes of antibody that can be produced. Now, when they have to come out, so initially we how do I add the just delete this for a while. Hmm. So initially, when they are bound on the surface of the cell membrane, they are all produced in a Y shaped in its usual Y shaped structure. But when it comes into the circulation, we have two antibodies that have the ability. So we have two antibodies which can form secondary association with similar antibody of its own type. Which of these two? IgA, so which is secreted as the dimer with a J chain and a secretory component, whereas IgM is secreted as a pentavalent structure. So this pentavalent structure in here, we have five antibody molecules, five J chains, five IgM molecules, all of them joined together by disulfide linkages, and we also have a J chain that keeps the entire structure together. So we have, this is essentially how a IgM molecule looks. So in the red, we have disulfide bonds that are keeping all the pentameric peptides together. And there is also one J chain that can link just two of these pentamers together. And if this J chain is removed, essentially then this entire pentameric structure is broken down, and you have five different antibody molecules coming in the circulation. So this is once your bound receptor is released in the circulation from the plasma cell, it can assume a structure like this. Now, what is the purpose of it? First, the cross-linking by antigen becomes easier, because if you imagine that we have two antibody molecules like this, but if you just fold the structure, so they become, the antigen binding sites become closer, and it is more so structurally it benefits the individual by making it more feasible for the antigen to cross-link these two, and faster the immune response can be generated. So yes. Apart from that, for IgA, so the antibody molecule that is secreted out from, so secretion in terms of the secretion that comes out in the lumen. So in lumen, say for your bodily fluids, if they can have antibody molecule, we will see how that happens. So your primary antibody molecule that is present in your secretory fluids is IgA. This is facilitated by the secretory component that is present in the IgA molecules. So the structure of IgA in circulation, it looks like this. So we have a secretory molecule that keep these two chains together in addition to the J chain. So the role of J chain is more so in just holding the molecules together. The secretory component is what helps for the antibody molecule to come out into the circulation. So it's the antibody that actually crosses the transepithelial membrane of your gut, or the one that comes in the breast milk, so that the newborn can be benefited out of certain antibody that is imparted to him from by the mother in itself. So this is the primary antibody that is present there. IgG is the most abundantly produced antibody, and because this is of the secondary immune response, and it has the strongest affinity for binding the antigen. So this brings us to two other terms, which is affinity and avidity. So we just discussed that. So affinity is something. So we can calculate the efficiency of binding. So affinity determines the efficiency of binding. So class switching, affinity maturation while we're talking, how is that possible? Because what is actually determining your binding efficiency is the sequence of amino acids that is present. So amino acid sequence is again determined at the genetic level. So while we're talking about affinity maturing, we were saying that some sequence of genetic material can say be mutated so that your amino acid specificity can further be enhanced, that can increase the binding efficiency of the antigen and antibody. So in terms of affinity, IgG is a molecule that has high affinity, right? And this is the best for bringing about effector functions. The C terminal for most of the most of the effector functions that we will discuss, you will see that the IgG FC domain is primarily mediating the effector functions. So IgG is important in that context, but avidity is the total of binding affinities. So while we were discussing about why IgM is the first molecule to be expressed on the surface, we just saw that the secretion of IgM uh, it has the capacity of forming a pentavalent structure. So when there is a pentavalent structure, here you have high affinity, but two binding sites and two antigen binding sites. Over here, if we talk about IgM, because it exists as a pentamer, it has 5 into 2. So two binding sites on each antibody molecule and five such antibody molecule. Together, it has 10 antigen binding sites, right? So avidity, if you say, is the sum total of all the binding affinities, which is present for the overall structure that the antibody is present in the circulation. So avidity is higher for IgM, and that's why the body finds it more wise to produce IgM molecule, which is readily stationed on the naive B cells. And when it has to first encounter an antigen, it quickly comes into play and it can very nicely take care of the pathogen. We have also discussed in terms of the primary and the secondary responses, right? So for primary response, we saw that the lag period is very high, is pretty longer compared to the uh, lag period in the memory response. So what also happens, let's say this is 0 and say 14 days. So by the day 14, you see a primary response. Now we are going to break this up further in terms of the antibody that is produced. So we have the IgM because it first comes into play, and just after this happens, there's a happening of class switching there, and IgG antibody takes over. When there is a subsequent infection, we say that the lag period has shortened. The IgM response, however, remains of the same amplitude, although it arises faster. It's the IgG response that is primarily responsible for the heightened immune response. This is IgG. and this is IgM. So if you compare the amplitude of, okay, I should draw this. The amplitude of IgM response is typically the same for your primary immune response as well as for the secondary immune response. It's the IgG molecules that is playing a major role for your secondary immune response. IgE, as we were talking, it only comes into play when there is some allergic response or if there is a say a parasitic infestation in the body. That is when IgE comes into the body into play. So more so, you will always have IgG and IgM molecules, which is prevalent in the blood. IgA is prevalent in the secretions of the body. IgE only comes into picture when there is a parasitic infection or a allergic response, or if in case of certain autoimmune responses as well. IgD do not have a lot of role to play, but we saw that during the process of development, IgD is formed. But again, if IgD is knocked down, there is no effect on the overall process of development of the B cells. So it's also present in very minute quantities in the circulation. So while we were discussing about the different types of classification possible based on the different chains there is also very very minor differences in the amino acid sequence of the gamma and alpha chain that is possible. So based on which we also have alpha 1 and alpha 2 classification possible. So this is just different classification that comes into picture because of differences in the antibody constant region of the antibody chains. And for gamma, we have five such possibilities. So we have gamma 1, gamma 2, gamma 3, and exactly based on whatever is the make of the antibody can be called as IgG5. So it's nomenclatured like this. Again, all the different antibodies have different prevalences and different function. I think the most predominant is the IG1 type, but I'm not sure. We have to check that. So I'll mention this. So this is a very quick overview of the different types of antibodies and how we are classifying that. I think we can stop here for today because I'll just check if I'm missing anything major here. Hmm. So for our next class, we'll probably start from the effector functions that the antibody molecule can mediate, and then we can discuss about how the diversity of antibody molecules are generated. Okay, so we have Naval who is still present there. So if there's anything that you have to ask Naval, you may go ahead, or we can close the session for today.
No, ma'am, it's clear till now.
Okay. So also, since the initial antibodies that comes into play is the IgM and IgD, we there's also a reference of calling the IgG, A, and E as the antibodies of secondary immune response. Okay, that is it. Okay, we'll end the session for today. I'll see you all the next class.